I presented research about how to model Wnt secretion in squamous cell carcinoma. The skin is quite a complex organ, and it turned out that for its continuous function there need to be several pathways to organize and fulfill the functions throughout lifetime. And amongst other signaling pathways, the Wnt signaling pathway plays decisive roles in controlling cell proliferation, differentiation, and also stem cell maintenance and fate decisions, and thereby ensures to keep the delicate balance between stem cells and stemness on the one hand and differentiation of those cells on the other hand. So besides that the Wnt signaling pathway seems to play an important role in the skin. There have already been publications studying this, but this is rather controversial as they point out either a canonical Wnt signaling pathway or a non-canonical Wnt signaling pathway which are quite the opposite of each other. So it's not quite clear what is ongoing in this type of cancer, and we would like to model that by a system that has been established by our lab, because we try to model that by a cargo receptor named EVI, who is important for the transport of all Wnt proteins. And by modulating this, you in principle can modulate the transport of all Wnt's and therefore globally affect Wnt signaling cascades. I have a cell line that has been generated in a Black 6 mouse model, and I did a CRISPR-Cas9 knockout of EVI in either exon 1 or exon 3 of the gene. And I further characterized those cells, expanded them as single cell clones, and then characterized them in vitro. Of course, validated that they do not express EVI on protein levels anymore, and also that they functionally do not secrete Wnt proteins anymore into to the serum when they are cultured in cell culture. No, because concerning those EVI knockouts, they basically are not able to secrete either the so-called canonical Wnt's that rather activate the canonical Wnt signaling pathway, nor the non-canonical Wnt's, so they are not secreted anymore. We wondered then how those cells differ in cell culture now that we have a knockout clone of the cell line that basically is a squamous cell carcinoma cell line that has been isolated from a mouse model that has been induced this cancer by chemical carcinogenesis and was then generated as a mouse SCC cell line. And I tested those cells in several experiments in vitro, and I found no difference between wild-type control cells and also EVI knockout cells. So they completely behave the same concerning proliferation. They do not have a defect concerning viability when you assess that, for example, with cell titer flow assay measuring ATP levels. And they also do not differ in their composition and cell cycle. So they seem to behave in vitro completely the same. Then we tried to inject those cells into Black 6 mice because they originally come from Black 6 mice, so this is working. And we saw that the EVI knockout cells are way less tumorigenic compared to the wild-type and control cells. So this is pretty interesting, and we are currently trying to find out what the reason for this might be by quantitative proteomic analysis and also by expression profiling.